So we were discussing receptor tyrosine kinases, and in today's class, we are starting this particular pathway. This is called RAS MAP kinase pathway, which is one of the signaling pathways downstream these receptor tyrosine kinases. Okay, and in addition, I would like to tell you this is not only downstream to these receptor tyrosine kinases, even some GPCRs are also sending their signals inside the cell via these MAP kinase pathway. So, okay, it is written here. So from here, we can find it out. These MAP kinase pathways refers to the cascade of protein kinases. So we know kinases, these are phosphorylating proteins. So they will take the gamma phosphate from the ATP and will transfer this gamma phosphate to some of the substrate proteins that are highly conserved in evolution, and they are present in all eukaryotes from yeast to humans. Okay, so the central element in the pathway are the family of proteins serine, threonine, or kinases, and these serine, threonine kinases are called MAP kinases. But before MAP kinases, there are another proteins which may be either. So first of all, this is important. They are serine threonine kinases, so they are MAP kinases. MAP for mitogen activated protein kinases. So there are certain chemicals. Okay, so these chemicals are called mitogen. Mitogen are okay. So in response of which the cell will start dividing, because these MAP kinases are one of the major signaling pathway within our cells. Okay, now, these are activated in response of variety of growth factors, which are called mitogens or other signaling molecules. So these can activate these MAP kinase pathways. And the second name for these MAP kinases is actually, they are called ERK. This pathway is called ERK pathway for extracellular signal regulated kinases, because the signal will come from the outside. For example, some growth factor will come from the outside. Okay, actually this is not the correct diagram to depict this particular thing, because this is mTOR pathway. So actually the MAP kinase pathway is given on page number 628 in your Cooper in chapter number 15. So this growth factor will come and bind to a receptor tyrosine kinase. Till now, it is clear that these receptor tyrosine kinases and GPCRs are forming the cell surface receptors to which the signal molecules or growth factor will come and bind. Then there are certain receptor tyrosine kinases domains inside the cell. These are called C-terminal domain of these receptors, which are having some enzymatic role or catalytic function. So they will phosphorylate the target proteins over their tyrosine residues. Okay, so if this is not clear from here also, so we can move one more page so that actually we will have another diagram which is more clear actually. So this particular receptor tyrosine kinase will act as, because they are second largest cell surface receptors with after GPCR proteins, so they will receive the signal on extracellular surface. So growth factor will come and bind to these N-terminal domain. Fine. So then this catalytic domain and the extra catalytic domains will be phosphorylated over specific tyrosine residues and will create a binding site for those proteins which are having some SH2 domains. So remember always, so only those proteins which are having these domains, and these domains are called SH2 domains, will be able to bind to these uh, phosphotyrosine residues, because the tyrosine residues are phosphorylated and the SH2 domain containing protein are able to interact with these phosphotyrosine residues. Now, this protein that is called GRB2 is an example of adapter protein. So I'm writing it adapter. So you need to note it down. What are adapter proteins? So uh, these are very similar to the adapters you use in your houses, at your home. These adapters are having some of the function. Okay, so these adapters are having some of the functions. Uh, do not know why this is not working. So these adapter proteins are having a function which connect the two proteins together. So therefore they are called adapter proteins. Now how they are able to work so efficiently in such a way, because they are very specifically interacting with both the proteins, because they are having SH2 domains. Therefore, they will able to interact with these receptor tyrosine kinases over their phosphotyrosine residues. So it is tyrosine here. So this tyrosine is represented by Y. You know the single letter codes. So tyrosine is Y. So Y will be phosphorylated. This P is phosphorylated form of Y. So it will bind to SH2 domain, whereas one another protein which is called SOS, and you know this particular protein is a guanine nucleotide exchange factor. So what is the function of the GEF protein? So they will transfer, or they will help in exchange of these GDP molecules with some GTP molecules, so that these RAS proteins can be activated. Okay, so, but actually one more thing I missed in this particular diagram is this particular domain. So these two particular domains are quite different from this particular domain, as you can notice. Therefore, you need to write here somewhere, these are called SH3 domains. Okay, so the GRB2 protein will act like adapter because it is having two SH3 domains and one SH2 domain. This SH2 domain will interact with phosphotyrosine, whereas this SH3 domains will interact with one another protein that is called guanine nucleotide exchange factor, which will help this RAS protein, which is a member of monomeric G proteins. So remember, when we started GPCR, I told you there are two types of G proteins. So these GPCRs are G protein coupled receptors. So they were interacting with some of the G proteins. Similarly, these receptor tyrosine kinases are also having some analogous proteins with which they are able to transmit the signal in the cell cytosol. So these are called, these are the family of monomeric G proteins. So in GPCR, actually, we have studied trimeric G proteins, which were having alpha, beta, gamma subunits, and the alpha subunit will be able to bind to the GTP. Similarly, these RAS proteins are analogous to the alpha subunit of trimeric G protein, because it is able to bind to GTP, and once it is bound with GTP, it will be activated. If it is bound with GDP, then it will be in inactive state. So hope this is clear. So once it is means this guanine nucleotide exchange factor transferred GTP over this GDP, it will be exchanged. GDP will remove and GTP will bind to the RAS, and see this will be activated, and then it will activate some of the downstream proteins, for example, RAF. So now we will move to some another diagram over the last page where we have left actually. So remember, this RAS and RAF are forming a complex, and this RAS, which is active form because it is GTP bound form, is activating another downstream protein that is called RAF. Now this RAF, okay, so this RAS is activating this RAF, and then this RAF is actually a kinase. Once it is activated, so I'm changing color and I'm writing here, this is either a serine or threonine kinase. Okay, so it will add some of the phosphate from ATP, that will transfer the gamma phosphate actually from the ATP to the serine or threonine of downstream protein called MEK, MEK, and it will phosphorylate either serine or threonine. Mostly it will phosphorylate the serine residues. So these phosphates, so here it is having some serine, or here it is having some threonine or threonine. So they will be phosphorylated, and it will result in the activation of this particular MEK. Now it will result in the activation of MEK. So once this is activated, this will another protein kinase, so it will phosphorylate either, okay, so this is not a serine kinase now, this will phosphorylate either threonine, so this is so color, so this is either threonine or it will phosphorylate a tyrosine, so this is why, okay. So remember, this MAC is a dual specificity kinase. It will either phosphorylate a tyrosine, or this is tyrosine actually, or threonine T in the downstream protein called ERK. And in ERK, the two amino acids are nearby, means at 183 position, there is a amino acid that is called threonine. So at, if this is 183, so this is called threonine. So threonine over 183 position. So remember these positions also, because actually these positions are very important, because this is threonine and at 185th position means there is a single amino acid in between. So some amino acid X in between, and it is followed by tyrosine Y at 185th position. So it is some another amino acid at 185th position. So this is called dual specificity kinase, because it will phosphorylate both the threonine at 183 position and tyrosine at 185th position, and it will result in the activation of this particular downstream protein ERK. Now, this ERK will act again as a kinase, and it will transfer the gamma phosphate from the ATP. Now, again, you need to remember this. It will act as either a serine kinase or a threonine kinase. So mostly in the cells are either serine or threonine kinases. Only few examples you will find in cell signaling where they are act like threonine or tyrosine kinases. They will not phosphorylate the serine at all. Okay. So now hope this is clear, and it will phosphorylate some other downstream proteins. So let me clear all these drawings first. So it will phosphorylate some of the nuclear cytosolic proteins in response, and then thus this particular signal will be moving. So this is the theme of RAS MAP kinase pathway. So let us study from the text, and you will understand this particular pathway in more details. So from here, actually, I'm starting. So page number 627, the MAP kinase pathways. So these MAP kinase pathways are mitogen activated protein kinase pathway, or this is also called ERK pathway, that is extracellular signal regulated kinase pathway. So there is a family of these kinases, so they will controlling this particular pathway. MAP kinase pathway, they are universal, they are conserved, highly conserved in evolution. Okay, in evolution, they are highly conserved, and they are found in all eukaryotes, including humans, because they are conserved in all eukaryotes, ranging from yeast up to humans. Now, the central elements in this pathway are family of protein, serine, threonine kinases. As we have seen, these protein, serine, threonine kinases are called MAP kinases. So if you remember from the last diagram, what are these MAP kinases? So this is ERK protein, as we have seen in this particular diagram. So this ERK is actually the MAP kinase. So you can write in your diagram also. So it will help you better understand. So this is called MAP kinase. MAP kinase. So if you remember, this is a serine threonine kinase, RAF. This is a threonine tyrosine kinase, and this is again a serine threonine kinase. So it will. So hope this is very understandable, that this particular proteins, either RAF, MAC, or ERK, all are kinases. So these are called serine threonine kinases. So in the downstream protein, they will phosphorylate either serine or threonine, mostly serine actually. MAC is a dual specificity kinase, so it will phosphorylate both. So at 183 position, it is a threonine. At 185 position, it is a tyrosine, and both will be phosphorylated to activate this particular MAP kinase. So therefore, you can call these also as kinases. So tell me if this is a MAP kinase, okay, so you can give the names to the remaining kinases. So this is called as MAP kinase, MAPK. And because this is MAP kinase already, and this is a kinase for this particular protein, so you can add one more kinase. So this is called MAP kinase. Similarly, this particular protein RAF is activating this particular kinase, so you may add one more kinase. So this is called MAP kinase. Already this is a MAP kinase. So because it is coming 2 positions earlier, so this you can add 2 more K. So actually this RAF, somewhere you will find that it is MAP kinase. So God knows how many kinases early in this pathway, because some of the pathways are still needed to be elaborated. These arrows are showing there are certain other proteins which we actually do not know. Actually, some protein may exist in between these pathways. Okay. So, but we have studied about these particular proteins. Any queries? So, because this particular RAF protein is a serine threonine kinase, so there are some growth factors or other, okay, so they will help in activation of this particular pathway. We are here somewhere. And these are called mitogen activated protein kinases, activated in response to the growth factors or some of the mitogens, which are some chemicals actually. In yeast, these MAP kinase pathways control a variety of cellular processes. You can note these help in mating in yeast. They control the cell shape and the spore formation, that is called sporulation. In higher eukaryote, including the humans actually, or in mammals, they are ubiquitous regulators of cell growth and differentiation. So whenever there is a growth in cell, or the cell want to differentiate in some specific kind of cell, these MAP kinase pathway or these proteins, you will find them there. They are doing signaling in some form actually. The MAP kinases that were initially characterized in mammalian cells belong to ERK family. Therefore, this ERK is called MAP kinase. So actually, you can write here also, this is MAP kinase, MAP, MAP, and 1K. So this is MAP kinase. This ERK was first characterized in mammalian cells. ERK, and this ERK was given a name because these extracellular signal regulated kinases, because one signal was coming from outside and they are regulated by this extracellular signal, and they were actually working as kinases, either serine or threonine kinases. So therefore, it is given the name in this way. Okay, so they are either serine kinases or threonine kinases. So, okay, sorry, something is happening. Okay, so let me annotate this. So they are either serine kinases or threonine kinases. Okay. So this ERK activation plays a central role in signaling the cell proliferation, because they are controlling the cell growth and differentiation. So whenever the cell want to proliferate or survive, this particular MAP kinase signaling is happening. Not only cell survival or proliferation, actually these are also responsible for cell death. Okay, so we will come to that particular portion. Okay, so they are also involved in cell death. If you want to see, it is given here. Okay, so this particular thing. So all these are MAP kinase pathways, and in addition to ERK, mammalian cell also contain the genus kinase, JNK, and P38 MAP kinases, and where this RAS-RAF MAP kinase pathway is involved in cell survival, differentiation, and proliferation, in some inflammation and cell death, other kind of MAP kinases, for example, Janus kinase and P38 are involved. So not only cell survival, these pathways are important because they are also involved in cell death and inflammatory responses that are in immune responses actually. Now the question is, so here, so let me annotate. So there is a query. I do not know who's asking this, but let me see the query first. They're asking, sir, what is the position of CN3 in MAC and ERK? In MAC and ERK, actually, it is not given an importance. So obviously, but there are some specific positions. Okay, this is not mentioned, and this is not necessary actually. Whatever is urgent and necessary from the exam point of view, I will let you know. Okay, so actually, this is not mentioned here. Sorry. Okay, so now the question is how this signaling is happening. So, okay, so this extracellular ERK activation plays a central role in signaling the cell proliferation, and we have seen they are also involved in inflammatory responses as well as in cell death. And this is induced by certain growth factors or some type of signal molecules which that act through either protein tyrosine kinases and GPCRs actually. So that I want to tell you actually. So not only receptor tyrosine kinases, though we are studying this in context of receptor tyrosine kinases or protein tyrosine kinases, but these are also downstream pathways to some of the GPCRs actually, so G protein coupled receptors. And actually this protein kinase C we have studied, this C was activated, so some of you might forget this protein kinase C. So let me finish this particular text and then I will tell you how this protein kinase C was activated. Actually, this we have studied in IP3 DAG pathway where the protein kinase C was directly binding with the DAG, diacylglycerol, and some of the calcium ions will be coming and activating this protein kinase C. Now, what is the function of this protein kinase C? It can also activate this particular MAP kinase pathway or ERK pathway, and both the calcium and cyclic AMP from the GPCR signaling pathway intersect with ERK signaling. Because this particular ERK signaling can happen downstream to either protein tyrosine kinases or GPC. Therefore, either calcium, because this protein kinase C will be activated in response to calcium, and cyclic AMP will be produced in response to GPCR signaling. So they will intersect somewhere this particular MAP kinase pathway. Therefore, this is very, very important because this is coming in both GPCR signaling as well as in receptor tyrosine kinase signaling, either stimulating or inhibiting. And then this is always not necessary that it is stimulating something. It is also possible that it is inhibiting the ERK pathway in different types of cells. So depending upon cell types, it is completely flexible that whether it is stimulating or inhibiting the ERK pathway, or whether it is involving in the cell differentiation and proliferation or in cell death. This is always depend upon the cell types. So it can vary from cell to cell. So you need to remember this. Now, the question is how this protein kinase C was regulated. So from our previous discussion and classes, you should remember that. Okay, so this is called IP3 DAG pathway, where this PIP2, which was a minor component of your plasma membrane, was broken down from here and was converted into diacylglycerol and IP3. So this IP3 was a polar molecule, and it will be going and binding to the endoplasmic reticulum membrane, so that it is able to open some of the channels in endoplasmic reticulum membrane, so that the calcium ions will come in the cytosol. Okay, then what is the function of this diacylglycerol? This will be able to directly bind with some of the protein kinase C. So it is given here actually. Okay, So this PIP2 with the help of PLC beta gamma, beta and GPCR signaling, gamma and receptor tyrosine kinase signaling will be converted into diacylglycerol and IP3. This IP3 is opening some of the calcium ion channels in endoplasmic reticulum membrane, and this protein kinase C that is written here as protein kinase C, PKC, will be activated in two ways. One, this diacylglycerol, which is interacting with the plasma membrane, will recruit this protein kinase C from cytosol and at the plasma membrane, and it will activate this protein kinase C. Second thing, this calcium ions will go to the protein kinase C and will able to activate the protein kinase C. Then this protein kinase C is a serine threonine kinase again, and it will be able to recruit or activate many of the proteins in the cytosol, which are their target proteins or effector proteins. So this particular term also you need to remember. These effector proteins are some downstream proteins which will be activated in response to that particular kinase. For example, here protein kinase C. Okay. Now, actually all this we have discussed. So we are moving back to our topic of interest, that is MAP kinase pathway or ERK pathway. Now, the activation of this particular MAP kinase or ERK is mediated by two upstream protein kinases. One is called this particular, which is coupled to some of the growth factor, that is called RAS. RAS is a GTP binding protein, and activation of RAS lead to the activation of RAF. So if you are not able to get the points, everything is given in form of diagrams. So some growth factor will come, will activate the RAS. This RAS will activate the RAF. And this, you know, that this RAF is actually interacting with this particular receptor as I showed you. So from here actually you will find this. So this RAS is actually interacting with not directly but indirectly with this receptor tyrosine kinase with the help of this adaptor protein and this guanine nucleotide exchange factor, which is helping in the activation of this particular RAS. Now, question is how this is regulated. So it is regulated with the help of this guanine nucleotide exchange factor. First of all, this guanine nucleotide exchange factor will activate this RAS by transferring GTP, and this GTP will be released, thus it is activated. Second thing is some gap proteins which are called GTPase activating protein. So this RAS, which is able to bind with this GTP, is having some intrinsic GTPase activity, but this GTPase activity is in the control of some of the other proteins which are called GTPase activating proteins. So some GTPase activating proteins, or these are called GTPase activating protein, GAP proteins. So these gaps will activate this RAS GTPase activity, so that this bound GTP will be hydrolyzed to form GDP again, and this GDP will bind with this particular RAS and it will be inactivated. So that's how it will be regulated. So with the help of two proteins, one is GF, which will activate this. The gap proteins will help in inactivation of the RAS protein. So this RAS is followed by one downstream protein that is called RAF, then other proteins will come into the picture, and that's how the signaling is happening inside the cell. So for example, you can also see from here these gap proteins, how they are functioning. So this is RAS, which is interacting with GDP, and this is the inactive state. Then the GF, that is guanine nucleotide exchange factor, that is some specific protein. This GF is actually called SOS protein. Okay, so this SOS protein will transfer GTP and will release GDP, so that the RAS protein will be activated. Then other protein will come, that is called GAP, that is GTPase activating protein of this particular RAS. So RAS is GTPase itself, but it will not hydrolyze GTP until and unless this particular GAP protein come and tell him to do so. Okay, so this GTPase will be activated, and then this inorganic phosphate will be released out of this GTP, so that the active form will be converted into an inactive form, that is called GDP bound form. So this active and active form is a kind of regulation which is happening in this case, regulation of RAS proteins. Then Then this RAS, RAF, MAC, and ERK, they are, this is MAP kinase, this is MAP kinase kinase, this is MAP kinase 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 actually. So they are, actually we are moving from this towards this particular thing. So actually the signaling is happening in this way, but because our central regulator of this pathway is this particular MAP kinase. So we are moving from here to this particular regulatory molecule, then this particular regulatory kinase. Okay, so we are moving from ERK to MAC to RAF. So now, the central role of this particular pathway in the mammalian cell emerged from the studies of RAS protein. We do not know anything, actually, we were not knowing anything, but actually in 1982, the things start changing. Means what will happen, what was happen actually. So if there is some mutation in this particular gene that is called RAS gene, okay, so this RAS gene is, this is for rat, and this S for sarcoma. So this is a rat sarcoma. So in rat, there's one kind of tumor that is called sarcoma is seen in 1982, and it was because of mutation in this particular gene. Okay, so it was first identified that the oncogenic protein of tumor virus, so some tumor virus is having some oncogenic or tumor inducing proteins that causes sarcoma in rats. This is called RAS. Okay, so therefore the name came as RAS for rat sarcoma virus. Interest in RAS was intensified, means much research was done after 1982, because when the scientists seen that this mutation, even a single mutation in this particular RAS gene, which was creating the sarcoma in rat, it can be implicated in the development of cancer in the humans. So if something is causing cancer in rat and same thing is creating cancers in humans also, so that is something very interesting actually. So scientists started reading about this particular gene, which is called as RAS. Okay, so this RAS gene mutation is creating some protein with mutation, this protein RAS. If it is mutated, so you know what is the function of RAS? So RAS is some protein which will be activated when it is bound with GTP. So suppose if there is some mutation in RAS, so that it will preferentially bind with GTP, so will remain in an active state for prolonged period of time. तो actually होगा क्या, अगर RAS प्रोटीन में कोई म्यूटेशन हो जाए तो वो कंटिन्यूअसली GTP के साथ इंटरेक्ट करती रहेगी, और उसका जो GTPase डोमेन है, जो GTP को हाइड्रोलाइज करने वाला डोमेन है, वो प्रॉपर्ली वर्क नहीं करेगा. अगर वो बहुत ज्यादा टाइम तक एक्टिव फॉर्म में रहेगी तो जो पाथवे है, जो मैप काइनेज पाथवे है, it will be active for prolonged period of time. So this signaling will continue to happen. Okay, if this RAS protein is active, then the signaling will continue to happen, and many of the nuclear and cytosolic proteins will be activated, so that it can lead to the cell proliferation in such a way that can lead to tumor or cancer. So therefore, it was very interesting when it was reported that around 1982 that it is also can be implicated in human cancers. Importance of RAS in intracellular signaling was then indicated by the experiment, which was showing that microinjection of active RAS protein directly induces proliferation of normal mammalian cells. Now, the two type of experiments were done. In one experiment, these RAS proteins, which were active actually, means which were actually bound with GTP. We know this GTP bound form is active. If you give a microinjection, so if microinjection is given to some animal or some mammal, so a normal mammal showed proliferation of his or her cells. Okay, so it will lead to the proliferation of cells. Second thing is, so this was the first thing. So this microinjection was the first thing which was recorded, which was reported. Second thing is interference with RAS function by either microinjection of anti-RAS antibodies. So suppose if you are injecting some antibodies against this particular active RAS protein, so what will happen? these antibodies will neutralize the active RAS protein, so that RAS will not be available in the cell to do proliferation signaling. So the cell will not proliferate at all. Or you will create some genetic dominant negative RAS mutants. You created some mutants which will block growth factor induced cell proliferation in all the cells. So thus the RAS protein is not only capable, so they were able to recognize that this RAS protein is not only capable of inducing the abnormal growth proliferation of cancer cells, but it is also appeared that it is important, it is required for the responses of normal cells to growth factor stimulation. So if the normal cell want to proliferate, they also require the RAS protein. And if the RAS protein is in excess or it is activated for a longer period of time, for example, some RAS protein active microinjection is given to some animal or mammal, so it will show uncontrolled proliferation of those cells. So it will be causing tumor or cancer. So both ways are important. Okay. But you cannot delete this particular RAS gene because this is required for normal response in normal cells proliferation, because this is urgent. Now the next question was, what is the nature of this particular RAS protein? So actually we have seen this. Okay, Shreya is asking something. When this RAS protein is activated, okay, so this RAS is activate the RAF protein, yes. Then RAS use GTP for activate RAF. Yes, because the RAS in its GTP bound form is active state actually. So if you remember Shreya here, so see, once this RAS is bound with GTP, only then this is activated. And once it is activated, this will activate the downstream protein that is called RAF. Okay. So this will interact with RAF and it will also activate this particular kinase. Fine. Now we are moving to another discussion. So now the question was, what is the function? Sorry, after function, what is the structure of this particular RAS protein? So from our discussion now, it is clear that this RAS protein is going to bind some of the GTP. So this is a guanine nucleotide binding protein. So because it is bound, it will bind either GTP or GTP or GDP. So in both ways, it is either bound with GDP, and this is the inactive form. And when it is bound with GTP, this is the active form. So this is some guanine nucleotide binding protein. So already bound with GDP, and if it want to activate, this will bind with GTP with the help of some SOS protein or some guanine nucleotide exchange factor present in your cell cytosol. So, and it is very analogous. So it is also important to note here that it is very analogous to the alpha subunit of G protein. So in G proteins, we have studied three subunits, alpha, beta, and gamma. And you know this beta gamma is forming a complex, whereas this alpha is bound with either GTP or GDP. And when it is bound with GTP, it will be dissociated from this beta gamma complex and will activate other proteins. Similarly, this RAS protein is behaving like alpha subunit of G protein, as I have already told you about this particular. Okay. So it will alternate between the active or inactive form. So inactive GDP bound and active form. So in contrast to the G protein alpha subunit, however, this particular function, RAS functions as a monomer rather than in association with beta gamma. So you know that the G protein is also having this beta gamma subunits, but this RAS will function as a monomer. It will not have these beta gamma subunits. So this is clear now. RAS activation is mediated by a guanine nucleotide exchange factor, that is GEF, some protein that is actually, when was first recognized, it is named as SOS protein. So this SOS is actually our guanine nucleotide exchange factor that will help in the activation of this RAS protein so that this RAS protein can interact with GTP. instead of GDP. And the second thing you need to note here, this GTP will be fresh, means it will come a new GDP. It is not like the GDP will be converted into GDP. It is never happening. The GDP will be replaced by the GDP because the cell is having high concentration of GDP. So GDP can be easily replaced by GDP molecule and will help in activation of RAS protein. Now, this exchange factor, which is a SOS protein, actually, the activity of RAS GTP complex is then terminated by GTP hydrolysis. So somehow, if you remove this GTP and convert this GTP into GDP, that is called GTP hydrolysis. And this GTP hydrolysis will leave RAS protein inactive. Okay, so who is going to hydrolyze this? So some GTP is activating protein that is called GAP. So this GAP protein is going to this is called GAP. So this GTPase activating protein will hydrolyze bound GTP and will convert GTP into GDP so that this RAS will be inactivated. It is interesting to note. Now, this point I want you to note because this is also very important. If there is a mutation of RAS gene, because this RAS gene is able to bound to both either GDP or GTP. Now, if there is a mutation in human cancers, it was found that have the effect of inhibiting GTP hydrolysis. So this GTPase activating protein will be not able to activate the GTPase domain of RAS protein. So inhibiting the GTP hydrolysis, the bound GTP will not be hydrolyzed. And if it is not hydrolyzed, it will remain in an active state for a prolonged period of times. Okay, so these mutated RAS proteins which were bound with GTP, therefore remain continuously active GTP bound form. Okay, so they will remain in GTP bound forms, driving the unregulated proliferation of cancer cells, and therefore they are creating cancers in humans. Even in the absence of, even in अगर मान लो कोई growth factor नहीं है to stimulate the signaling, even then they are able to signal. Therefore, there is uncontrolled proliferation in the, even in absence of a growth factor stimulation. This is very important to know. So after you get this, the RAS protein are prototypes of large family of approximately 50 related proteins. So in the RAS protein, there are about 50 proteins which are given in your pathfinders. So let me show you these. Okay, so here also actually we have annotated. So here all triangles and then. So see here. Okay, so. Okay, so this monomeric G proteins, which are analogous to this particular RAS protein, there are related families. One is called RAS, which regulate the cell growth through the serine threonine protein kinases. As we have seen, Rho family is very related, and Rho, RUB, RAN, SAR, ARF. So they are all related families. So they are having some 50 proteins which are involved in either cell growth, regulation, cytoskeletal reorganization, vesicle targeting fusion, vesicle formation in cells so that the cargo can be transported from one location into the another in the cell cytosol, or function in the transport of RNA and protein out of the nucleus. So they are having different functions, but they are coming under the same family. Okay, so they are approximately 50 related proteins, so frequently called as small GTP binding proteins, because RAS and its relatives are about the half size of G protein alpha subunit, because they are very related. They are analogous to alpha subunit, but their sizes are small. They are about half size of this particular G protein alpha subunit. And this is also important to underline that this RAB, which we have already studied in our mTOR pathway, it is also a member of this particular family. One member of this family is called RAB, which regulates the mTOR signaling, which we have studied in our discussion. So if you do not remember the RAB protein, then I will show you this. So this is a RAB protein, and this RAB protein is controlling your mTOR-raptor complex. This is also a GTP binding protein, and in this case, actually, this is GTP binding protein, and the earlier complex, which is formed by the TSC1 and 2, this is called GAP. GAP is GTPase activating protein. So in this case, this complex is GTPase activating protein. It will help this RAB to either activate or inactivate. When it is bound with GTP, this will be activated and this signaling will happen so that protein synthesis can happen, translation will happen. And if it will stimulate the GTP so that the bound GTP is hydrolyzed, so GTP will be converted into GDP and the RAB will be inactivated so that the protein synthesis or translation will not be happening in the cell. Right? Now, this particular finding or this results are given in your book. This is showing that in so it is showing that the most of the cancers are means in America in developed countries, for example in USA. So they are causing all about 1/4 deaths. Means about one individual out of four will die out of cancer. And these cancers are actually for an year, if you count, they are counting for about 6 lakh deaths in each year in the US. So, if you go into the statics, so most of the cancers are colon or rectum cancers, followed by lung cancers, and the lung and these two type of cancers are accounting for 40% of all cancer deaths in the US. Okay, rest are either breast cancer, pancreas, prostate. So these are counting for 7.2, 5.6, and 5.3 respectively. And these calculations and how this was identified, how the RAS gene was identified in 1981 or 1982, is everything is given in this particular text. So if you're having this particular book, you can read it. Okay, so hey, we have studied this. These will be activated, and that's how they will control many aspects of cell signaling. Now, from here actually, so the best understood mode of RAS activation, as we know, because it is mediated by receptor tyrosine, protein tyrosine kinases, though we also know that they will be activated in response to GPCRs, but because most of the research is done in this particular domain, we know more about this. And actually, the research is a result of people's interest. Okay, and most important is funding. So if scientists got the funding in this particular domain, so they will achieve more better understanding and result in this particular way. Okay, so this protein tyrosine kinases are more understood. Okay, so autophosphorylation, because these protein tyrosine kinases are tyrosine kinases, and there is auto cross phosphorylation will be happening to activate the receptors or the catalytic domains. These receptors result in their association with the RAS guanine nucleotide exchange factor, that is as a result of SH2 mediated protein interaction. One well characterized example is provided by this protein, that is a guanine nucleotide exchange factor. This is called SOS. So as you show here, as you see here, this SOS is interacting with this receptor tyrosine kinase with the help of one protein that is called adapter protein, that is GRB2. And this GRB2 is having two kind of domains, as we have discussed, SH2 and SH3 domains. So everything, only these things are being discussed in this particular text. Now, this tyrosine phosphorylation of the receptor or other receptor-associated proteins create a binding site or docking site for the GRB protein, and this GRB is having some SH2 domain. So initially, actually, what will happen? This association of GRB2 with the activated receptor localizes SOS to the plasma membrane. So this GRB2 is adapter, and this GRB2 is interacting with the SOS, so it will localize SOS to the plasma membrane, where it will interact with the receptor tyrosine kinase. Okay, so where it is able to interact with the RAS protein also. because the RAS protein is already interacting with the plasma membrane. So this is the plasma membrane, and this is the receptor in your plasma membrane. So uh, the protein need to be localized so that the response is more effective. So this GRB2, which is adapter protein, is having SS2 domain, and it is interacting with some guanine nucleotide exchange factor, will recruit this GF from the cytosol at the plasma membrane, where the RAS protein is already interacting with the plasma membrane with the help of its C-terminal domain, CTD, so that these protein can come in close proximity and can activate each other. Okay, so this will activate this. Now, okay, so the RAS C terminal is actually anchored to the plasma membrane, inner leaflet of plasma membrane. So it is written here, so you can imply inner leaflet of plasma membrane by the lipid and attached by the RAS protein C terminus. Then this SOS, which is a guanine nucleotide exchange factor, stimulate the guanine nucleotide exchange, resulting in the formation of an active RAS GTP complex. In this active form, RAS interact with a number of effector proteins. So you need to remember these effector proteins. What are the effector proteins? So the proteins which are under the direct control of this particular RAS are effector proteins for this RAS. Okay, so for example, RAF is its effector protein. So including the RAF protein, which is a serine threonine kinase. And this interaction with the RAS recruit the RAF to the from the cytosol to the plasma membrane and initiate RAF activation, which also involves the phosphorylation of RAF by both protein tyrosine and protein serine threonine kinases. So this RAF protein will be activated in the response to these phosphorylations, which by both either tyrosine kinase or a serine threonine kinase. So RAF will be activated. So actually, sorry. Okay. So see, this RAS protein is interacting with this guanine nucleotide exchange factor, and then this RAS, after activation, after bound to the GTP, will be activating the RAF, and in this process, RAF will be phosphorylated at several positions. Okay, so either serine or threonine or the tyrosine kinases will help. Actually, it is not known which of the specific residues are being phosphorylated. Okay, so they will be phosphorylated, and this overall result in the activation of the RAF. So then this RAF will activate some MAC protein and so on, ERK. So, okay, here. So this RAF will activate MAC will ERK, MAC then phosphorylate ERK, and then some of the nuclear and cytosolic protein will be activated. So okay, so this RAF recruited from the cytosol into the plasma membrane and thus will be activated. As already noted, activation of a RAF initiate a protein kinase cascade leading to the ERK activation. ERK phosphorylate variety of target proteins, which are including some protein kinases. In addition, ERK regulates the mTOR pathway by phosphorylating this particular. Okay, so not only this, actually, this is also regulating this ERK, or this is called MAP kinase, phosphorylate a variety of target proteins, out of which one is TSC2, which we have already studied in our mTOR pathway. So it is somehow regulating this mTOR pathway also. And in this regard, importantly, fraction of activated ERK translocated to the nucleus. Once this is phosphorylated, so this MAP kinase or ERK protein will go inside the nucleus, where it regulates the transcription factors by phosphorylation or and other proteins. In this regard, it is notable that primary response of growth factor stimulation is a rapid transcriptional induction of family of about, so there is a family of about 100 genes, which is called IEG, immediate early genes. Sorry. So these are called IEG, that is immediate early genes. And in this family, there are about 100 genes which are under the direct control of this ERK. And you know, this ERK is your MAP kinase from our previous discussion. So this MAP kinase will be phosphorylated and will move inside the nucleus where it will phosphorylate number of proteins and transcription factors. Okay, so see here, it is going inside the nucleus and it will be phosphorylating number of proteins, for example, ELK1 and SRF. Now we will see what are these ELK1 and SRF. Okay, and this is the response element where this particular DNA response element is the DNA where these protein will interact and will control the transcription of these early genes, about 100 genes under this control. Okay, these are called immediate early genes, and the induction of number of immediate early genes is mediated by regulatory sequences which are called serum response element, that is SRE. So this SRE is serum response elements where these serum response factors will bind. So these are called serum response factor SRF, and the another protein or the transcription factor that is called ELK1. ERK phosphorylate and activate ELK1. This transcription factor will be activated, and thus the transcription will be started, providing the direct link between the ERK family of MAP kinases and immediate early gene induction of transcription. Many immediate early genes in themselves encode the transcription factors, so their induction and the response to the growth factor stimulation lead to the altered expression of battery of other downstream genes, which are called second response elements. So now you have two terms. One is called immediate early genes, and these immediate early genes will then cause the activation of second response genes, which will be downstream genes of these immediate early genes. So after all, there is a change in the expression, or cell will proliferate. Okay, so the cell will proliferate, divide, and will differentiate. As discussed in this chapter 16, actually, these alterations in the gene expression directly linked with the ERK signaling to the stimulate of cell proliferation induced by growth factor. So a growth factor is able to proliferate the cell with the help of this ERK pathway, or this is called MAP kinase pathway. Okay, so before telling you about these cell death processes, I would like to tell you, okay, so this is given in your Pathfinder actually. Okay, so, okay. So this MAP kinase pathway, which is called ERK pathway, is activated in response to receptor tyrosine kinases, which are surface receptors, cell surface receptors, and may itself trigger multiple pathways. So as we have seen, out of which one is the MAP kinase pathway, which is the major pathway of cell differentiation and cell proliferation. So it is involving some second messenger molecules. The activation of RAS triggered by the activated receptor tyrosine kinases is short-lived. Okay, so this RAS protein activates a downstream serine threonine phosphorylation cascade that is called MAP kinase, for mitogen activated protein kinases. Activated RAS induce kinase cascade, and many of the kinases are activated in this way. So from here you will be able to understand the same thing. This RAS, when bound to the GTP, is active. This will activate RAF, and this RAF protein is a serine threonine kinase, which is called MAP kinase. Actually, this is 3K. MAP kinase. Then this will activate the MAC. MAC is, this is a tyrosine threonine kinase. And actually, the other name for this MAC is MAP kinase and one more kinase. And finally, there is MAP kinase. This is called ERK actually. Was first identified in mammals. Therefore, this is called ERK pathway, which is a serine threonine phosphorylation, or this kinase. It is also given here. This is serine threonine kinase. This is tyrosine threonine phosphorylation or kinase. So this is serine threonine kinase. So overall, there is a change in protein activity or change in the gene expression by some transcription factors, for example, ELK1, as we have discussed, or SRF, serum response factors. And some of the early genes and late genes will be activated and thus will change the cell fate. So it is also given that activated RAS will bind to the N-terminal domain of RAF, RAF is a MAP kinase, which is a serine threonine kinase. So RAF directly activity, the RAF activity will lead to the activation of MAC. MAC is a MAP kinase. So it will phosphorylate MAC, which is activated by phosphorylation of two serine residues. So you need to know this. It is the two serine residues which will be phosphorylated by RAF and MAC. It is unusual enzyme because there is dual specificity. It can phosphorylate both threonine and tyrosine and the target that is called ERK or the MAP kinase. The MAC kinase or MAP phosphorylate and activate the MAP kinase. And finally, the MAP kinase is a serine threonine kinase. So this all things we have discussed actually from the book Cooper. Okay, so you can read this. Then there is another family that is called Rho family. And this Rho family is also having two other proteins that is called RAC and CDC42. Okay, they are also involved and they are also the GTP binding proteins. Okay. Now the one more thing which is left in this pathway is beside this MAP kinase pathway, which is MAC signaling actually, there are another type of proteins which are either called J and K, that is Janus kinase, and P38 MAP kinases. So you are able to understand this cell survival and proliferation or differentiation pathway by which is under the control of some growth factors. Okay, so these growth factors are able to activate the RAS protein. RAS is a GTP bound protein, and this RAF protein will be activated. RAF will activate MAC. MAC will activate ERK, which is a MAP kinase, and this MAP kinase pathway will able to do proliferation, differentiation, or will help the cell to survive. On the other hand, there are other type of kinases, either J and K and P38, which are also MAP kinases actually, but they are involved in inflammatory responses or in cell death. They are under the control of either if there is either there is a stress in the cell, okay, either the inflammatory cytokines will come, and if there is a cell stress, under the stress conditions, for example, under high pH or under ultraviolet radiations or under heat conditions, the cell will activate these family of proteins, which are RAC, Rho, or CDC42, which are very analogous to RAS, but they will function in different pathways. So they will lead to the cell death or inflammation actually. So from here, actually, the Rho family is having Rho itself, RAC, and CDC42, which is given in Pathfinder, and they are having the different signaling mechanism. These RAC, Rho, and 42 are able to activate some of the proteins, for example, ASK1, MEKK, MEK kinase, or MLK1. This is also a kinase, or this kinase, TAK1. So different kinases will be activated analogous to RAF, and then it will activate some other kinases. So there is a cascade of events which will finally activate these MAP kinases. So these ERK, J and K, this is Janus kinase, and this is P38. These are MAP kinases actually. These are real MAP kinases, whereas these are called MAP kinase, and this is MAP kinase actually. And these are GTP bound proteins. So this will lead to the cell inflammation and death. So now it is clear that not only cell survival or proliferation or the cell differentiation is regulated by MAP kinase, whereas other type of cell death and inflammatory responses, which are immune responses, are also under the control of this particular pathway. Before closing today's class, actually, I would like to tell you about the adapter proteins. Oh, sorry, actually, adapter proteins we have discussed. So one example of adapter protein is this GRB2, which is having two type of domains, SS2 domains, which will interact with the phosphotyrosine, and this is having SS3 domain, which will help in interaction with SOS or this guanine nucleotide exchange factor. Similarly, beside adapter proteins, our cells also have some scaffold proteins. So see here and note these names actually. So I'm minimizing this for some time. So there are four proteins. One is called RAC, MLK, MKK7, JNK, and there is finally a response. So this JNK is a MAP kinase, This is MAP kinase. Okay, MAP kinase 7, and there is MAP kinase. So 3 times K. So this is called MLK protein. So under the control of RAC, so I'm annotating, means I'm highlighting these proteins. So see here, sorry, it's very sensitive actually. So see here, if I annotate, so this RAC is controlling this MEKK, okay, and it is controlling this MKK7 and this JNK, and finally there is a response. Okay, so these all proteins are bound to a single protein. Okay, so what is the name of that single protein? That single protein is called scaffold protein. So this scaffold protein is called GIP1. So this particular GIP1 protein, sorry, see this. This is called scaffold protein. So now what is the function of scaffold protein? Scaffold protein, because we know all these proteins are involved in single pathway of either inflammatory response or cell death. So this GIP1 is acting as a scaffold protein. Okay, the scaffold is recruiting all these proteins nearby so that they are able to activate each other. So this kind of response is a localized response, so that a battery of proteins can be activated to do a final response event or some inflammatory response or some cell death, for example. So therefore it is called scaffold protein. So this JIP1 is a scaffold protein. It organizes J&K MAP kinase pathway protein and its upstream activator MLK and MKK7 into a signal cassette, so that as a result of the specific association, so this is very specific because all these are involved in a single pathway. So this specific association with the help of this scaffold protein, okay, so activation of MLK by RAC lead to the specific and efficient, this is very efficient because they are very close interaction. So the efficient activation of MKK7, which is in turn activating the J and K. This is a MAP kinase actually, this is MAP kinase. So if the JNK is activated, so it will go inside the nucleus and phosphorylate many of the transcription factors, as we have seen in our previous diagram. So for example, here it is very clearly shown this ERK or the genus kinase will go inside the nucleus. It will phosphorylate some of the transcription factors. Not exactly same because these are involved in cell proliferation. And the second thing, these are involved in cell death. So obviously there are some different proteins, but they will work analogously. They will work similarly. So that's all for today. Actually, tomorrow we will start this JAK-STAT signaling or transforming growth factor beta and SMAD pathways. So these are another type of signaling pathways in our cells, and actually we are having very few pathways. One is more NF-kappa B signaling. This is also important. And some of the signals, for example, this hedgehog signaling, Wnt signaling, which is present in developing embryos. So Wnt signaling, hedgehog, and notch signaling. These are left actually. And with this, we will end our signal transduction. So hope tomorrow I will be able to finish the cell signaling, and then we will move to some other topic. Now, this is the time to look at queries. So, okay, so if you have query, Shrey is asking something. Okay, so that was answered actually. So if you are having more queries, you can ask.